When we look at muscles, we have to talk about contraction. Skeletal muscles are about 40% of your body weight. So if you think about how much you weigh, about 40% of that is part of your skeletal muscle, which is muscle itself, and then the connective and neural tissues or nerve tissues. So all of that together makes up about 40% of your body weight. And what we need it for, of course, is to move around. We're going to focus now on muscle fibers, which are the actual contraction point for your muscle. If a muscle is going to move, it has to contract and relax. In order for it to do that, we actually have a system set up that's going to allow us to use what we call the sliding filament theory. And what this means is we're going to use muscle filaments known as myofibrils, and we're going to allow them to move past one another to actually contract our muscles. So your focus for us is going to be on thick filaments, and I'm going to show you these in a minute, and thin filaments. They're different. They're thick and thin based on what they're made of. Thick filaments are made of myosin. These are proteins. It's muscle. And thin filaments are made of actin. So when we look at taking a muscle and breaking it down into the muscle fibers and then into a single muscle cell, what I'm actually interested in is something known as a sarcomere. This is the contraction point of the muscle. So this is our fundamental contraction unit. When someone asks you what's contracting on a muscle, it's the sarcomere. It's what actually gives the striated appearance to skeletal muscle. When we talk about skeletal muscle as being striated, this is why. It literally has lines on it, and you see that here. So when we look at a myofibril, what we're interested in is from this spot to this spot, the sarcomere. Well, what's so exciting about this thing? When we look at it up close, you actually see, and this is an SEM image of a sarcomere, you see that we've got a lot going on. We've got these lines that form, and that's what forms our striations on our muscles. But we also have this kind of dark area in the middle and these lighter areas on the outside, and they literally are labeled light and dark band based on that. But what we're interested in is the blue being the actin or the thin filaments, and the pink being the myosin or the thick filaments. And the whole job of muscle contraction is to make this distance, the distance between what we call the Z lines, so the Z line distance decreases when muscles contract, and it increases when a muscle relaxes. We do this, of course, all through ATP. So the sliding filament theory is that we actually have the thin filaments are going to slide past the thick filaments, all powered by ATP. So what are we doing? So remember, the blue is the thin, the pink is the thick. The thick is myosin, the thin is actin. So we are literally going to take these thin filaments, this middle part, the pink dark band, the dark band is the myosin or the thick filament. That never changes. This does not change. What we are going to change are these blue lines. And what we're going to do is pull them into the center. So we're going to get them to move into the middle. And you see that with every step here, these get a little closer until you finally get this overlap in a fully contracted sarcomere. Or you would think about it as a fully contracted muscle. And when we look at this contraction, we have to figure out how it's actually going to move. We know when a muscle contracts, it gets shorter. So we need to shorten this. Well, we're going to shorten it by decreasing that space or that distance between the Z lines every time. How are we going to do it? We're going to do it with something known as a myosin head. So myosin is part of the thick filaments. And what we're going to do is have the myosin heads actually attach to the actin and literally pull it like you're pulling on a rope. This is going to allow us to pull those filaments into the middle. Now, this is all done at neuromuscular junctions. So we're looking at the interaction between your nervous system and your muscular system. This shouldn't be a surprise that you need nerves to move your muscles. But what we want to look at is where this myosin head, so sticking out from the myosin filament, these myosin heads are going to reach out and grab on to the actin. And they do that powered by ATP. So they literally have a receptor for ATP on the myosin head. And that receptor, it's going to act as an enzyme, is going to split that ATP into ADP and phosphate, and when it does, energy is released. And when we get energy release, that myosin head is actually going to snap back or pivot, and when it does, it pulls the Z line. It pulls that actin. So every time this head is going to reach out, grab, and pull, that actin becomes closer and closer in the center. Those Z lines come closer and closer together. Now you notice, I hope, that there are many myosin heads on a single myosin filament. And they're all going to work very, very quickly to get this muscle to shorten. Because if you flex your muscle, how long does it take you? Microseconds, right? This is a very quick response. And the reason it's a quick response is because of these neuromuscular junctions where we get the nerves coming in.